busy. I'm gonna introduce our speaker today. For those who haven't met me in person yet, I'm David Fox, and I was asked if I could introduce Jake, which I'm very happy to do in his talk today, as part of the process of his moving forward with potential nomination to being a full professor in the department. So this is his talk as part of that promotion process. Jake got his, and there will be a chat box quiz at the end of this, a one question quiz. So Jake got his BS in geology from the University of Arizona in 2003, and then completed his PhD in geobiology at the University of Southern California. And then went from there to a postdoc at Caltech, sponsored by the Agron Institute, which is a very prestigious postdoc. And then he joined our faculty in 2010 as an assistant professor, and was promoted to associate professor with tenure in 2016. He's got a number of honors on his CV, reflecting his sort of growing prominence in geobiology, broadly speaking, including as a McKnight Land Grant professor here at the University of Minnesota from 2013 to 2015. He has almost 50 publications in a wide range of journals covering prominent journals in earth sciences, in geobiology, as well as in microbial biology, which is a reflection of not only the diversity of Jake's own research, but the sort of interdisciplinarity required to do the kind of research he does, where it involves knowledge in various projects of deep time earth science issues, to growing microbes, growing and studying microbes, modern living microbes in the lab, to observing them and sampling them in their natural habitats at the bottom of the ocean, using the Alvin submersible. He has that diversity in his research interests and background is reflected in the sources of his research funding. He has grants, current and past, from the National Science Foundation, the National Institute of Health, the Department of Energy's Joint Genome Institute, the American Chemical Society, NASA Exobiology, the Simons Foundation, the Sloan Foundation, as well as the University of Minnesota. So a really outstanding record of external research funding, again, reflecting that breadth of topics that are covered in the research Jake does on interactions between microbes and Earth system processes. He has graduated or has advised 3 PhD students to the completion of their degrees, including one that I was a co-advisor with him. 6 master's students, and he's had a number of visitors and research scientists in the past and currently in his lab, and has several current students, as I think he'll maybe mention in this talk today. So I want 2 reminders before we get to the little quiz. One, after the talk, as is kind of our want now, I think we're going to reserve the first opportunities for questions for the students. I'll try to remember to say this again at the end of the talk, or maybe Jake can also. And if when we exhaust the student questions, we'll open up questions for everybody else, and you can open your mic or you can type it into the chat, either way. And as far as I know, there will be refreshments afterwards for those in or near Tate who want to join. As has been the custom now, that will be on the west side of Tate on the mall. And be kind of obvious where to gather, because you'll see people from inside of Tate on the outside of Tate. And so before I turn it over to Jake for his talk, Brimstone Bugs, Dynamic Geological and Biological Interactions Involving Marine Sulfur Bacteria, I have one question for you, and I'll give you a moment to think about it and type your answer into the chat to everyone. And that question is, what did Jake do? What was Jake's occupation before he went to the University of Arizona? What did Jake do before he was an undergraduate? Oh, well, apparently everybody knows this. Whatever, I don't even care. Yes, Jake. Yes,
Jake,
yes, Jake. Yep. Yes, that he
okay.
did that. Air Force. No, he was not in the Air Force, David. That you're gonna get Jake really riled up with that kind of thing. He was an
John got it
intelligence,
right.
wasn't really a spy. So yeah, Jake was a paratrooper in the 82nd Airborne, which is pretty amazing. Throwing himself out of perfectly good airplanes while they're in the air. So on that note, I'll turn it over to Jake to tell us more about his research.
Great. Thanks so much, David. Thanks for the excellent introduction. I really appreciate that. So today I have a couple of stories for you that are about sulfur bacteria and their interactions both with other organisms and also with geologic substrates. Um, Before I continue, I want to point out that much of the research that I'm going to share with you today is the product of work and ideas of Dr. Beverly Flood, who is a joint lead investigator on both the NASA grant and the NSF grant that support the work that I'm going to show here today. And also Mr. Dalton Lepridge, who's a PhD student working with us that has done really a fantastic job on many aspects of both of the kind of stories and projects that I'm going to show you here today. So, kind of the structure of my talk is first I'm going to tell you what sulfur bacteria are and what they do, a little bit about just kind of the background of these organisms, and then I'm going to kind of talk about two different stories or lines of research. One of them involving interactions with sulfur bacteria and how we think that they are responsible for dissolving carbonates that form at methane seep settings. And then finally, I'm going to talk a little bit about this discovery we made of bacteria that are living inside of other sulfur bacteria and some of the potential implications there for our understanding of eukaryote evolution. Okay, so first, what are sulfur bacteria? We call them sulfur bacteria colloquially in our as researchers of them, but we also kind of more formal names that you'll hear are sulfur oxidizing bacteria or sulfur chemolithotrophs, or if they're autotrophs, you could hear them referred to as sulfur chemolithoautotrophs, which is quite a mouthful. But what do I mean by that? Well, organisms like us that are aerobic respirers, we're basically using electrons that we get from our food, from organic molecules in our food, and we're passing them to oxygen. And there's a chain of redox reactions that occur that our cells are able to harvest and basically convert redox potential energy into a charge gradient that then is turned into ATP, and that ATP then fuels all the stuff that we do, like allowing us to talk and move and do all the things we do. Contrast that with something like a phototroph. These are organisms like plants and phototrophic bacteria that use the energy of sunlight to power a similar type of what's called electron transport chain, redox reactions that basically ultimately get converted into ATP. And finally, this other group of organisms, the ones that I'm really going to be talking about today, are so-called chemolithotrophs. And these are organisms that basically use inorganic chemical substrates as electron donors for their electron transport chain. And so organisms, specifically sulfur bacteria or sulfur oxidizing chemolithotrophs, they basically take electrons from hydrogen sulfide or other reduced sulfur compounds, and they pass them on to some electron acceptor. Some of them use oxygen, just like we do. Other ones can use other things like nitrate as alternative electron acceptors. But still, they're doing a bunch of they're converting redox potential energy into ATP ultimately. But here, instead of food, their electron donor is hydrogen sulfide, and that's why we call them sulfur bacteria or sulfide oxidizing bacteria. And these organisms are not to be confused with sulfate reducing bacteria, which are a totally different group of organisms, or there are actually many clades that do this type of metabolism, but they're different from the sulfur oxidizers. In this case, sulfate reducing bacteria, they convert sulfate into hydrogen sulfide. So their electron acceptor is sulfate, and that's becoming hydrogen sulfide. And their donor is not hydrogen sulfide; it's actually organic compounds like similar to us. They're heterotrophs, or that some of them can also use dihydrogen as an electron donor. So these are totally different organisms than the sulfide oxidizing bacteria, but you'll note that these organisms produce hydrogen sulfide as a waste product of their metabolism that then can serve as an electron donor for the sulfur oxidizers. So very often these organisms are found uh, coupled, metabolically coupled, although often typically they're spatially separated because these organisms are strictly anaerobes typically, and sulfur oxidizers inhabit more oxygenated or what we call microaerophilic, low oxygen settings, typically, generally. Okay, so these are our sulfur oxidizers, the ones that we're going to be talking about today, and I just call them sulfur bacteria, and that's what a lot of people that study them call them, sulfur bacteria. And I kind of have grossly kind of simplified their metabolisms. They are able to metabolize lots and lots of different sulfur compounds, and they make lots of kind of intermediate chemical species, and the different biochemical reactions that they can carry out are very complex. And this little cartoon just tries to illustrate some of that complexity that I'm not showing with kind of the previous diagram there. But they can use all sorts of different sulfur compounds, and many of them can also use organics and grow as heterotrophs as well. Sulfur bacteria can be, they have a variety of different kind of morphologies and lifestyles. They can be small, or they can be large. They can be motile or non-motile, so that means they can either move themselves in the environment or not. Some of them live in association with animals or other microbes, and they can live in a whole variety of habitats, from lakes and deep ocean environments, and high pH or low pH, lots of different settings. So here are a couple of examples of small and large sulfur bacteria. These Celeribacter pectoensis is one organism that I'll be talking about that's very small, like a micron in diameter, versus these really giant sulfur bacteria, which are the largest known bacteria that get up to a millimeter in diameter for one cell. And so this first organism I'm going to talk about with respect to some methane seeps. It's a small organism, small bacterium, and it can grow either on sulfur compounds, or it can also grow heterotrophically if you give it organics. And we isolated this organism from Gulf of Mexico methane seeps, and we sequenced its genome, and we have it in the lab, and we basically used it in some experiments that I'll tell you about in a little bit. So that's one of our characters in these two stories. And the other one is my favorite organism in the whole world, which is Thia Margarita Namibiensis, which means sulfur pearl of Namibia. And so you can see that these kind of chains, these bright white chains of cells here, they look kind of resemble a string of pearls. And so that's where the name comes from. And they were first discovered off of Namibia, which is a place where I do a lot of work here in southwestern Africa. And so these organisms are really the largest known bacteria. As I said, here's a picture of a fruit fly for scale, and these are individual Thia Margarita cells. So you can pipette these individual cells, and you can see them without a microscope. So you can see one Thia Margarita cell without a microscope. It looks really small, like a speck of dust, but you can see it. And these organisms, they use, as I said before, they use sulfide as their electron donor, and they use oxygen as their electron acceptor, or they can use nitrate as well. And these organisms, they actually have most of the volume of the cell. These huge cells actually contains a central vacuole that stores nitrate inside of it, that they use as kind of like a scuba tank or a reserve kind of electron acceptor, or place to store their electron acceptor. And here you can see that kind of in this dark space in the cell. And you can also see, here's this very nice kind of 3D image of a Thia Margarita cell showing that central vacuole. This is some very nice microscopy that Dalton did here. And these are the bacteria on the outside, and this little bacteria living on the outside of the cell. And then Here's the big giant Margarita cell with a vacuole inside of it. And we think that these organisms store this nitrate as kind of a reserve, something to breathe, uh, because they live in environments that are periodically anoxic and highly sulfitic. So these are upwelling systems where there's very high biological primary productivity, but also a high oxygen demand and very relatively shallow shelf setting, continental shelf setting. Here you can see kind of the water temperature. It's very cold here, representing the upwelling zone. And this is where we find these Thia And in these sediments, you have lots and lots of degradation of all of this biomass. Basically, you have sediments that are mostly a diatomaceous ooze. And so there's lots of respiration going on there, and there's lots of sulfate reduction. So there's lots of hydrogen sulfide being produced. And sometimes, often, these sediments are completely anoxic, and even into the water column, the whole water column could be anoxic. But this is actually like an open shelf. It's not like some restricted lagoon or something. These are just really, really rich sediments that are really organic rich. And so that's where these organisms live. And we think that they basically kind of wait out these periods of having no access to electron acceptors by storing lots of nitrate inside a vacuole. And they have lots of other storage inclusions as well. They store carbon, they store phosphorus, um, they store elemental sulfur, and they're basically really kind of, we think, dynamic mediators of biogeochemical cycling of sulfur, carbon, nitrogen, and phosphorus. So these are very interesting organisms from a variety of perspectives. So this is kind of the two characters that I'm going to be talking about today in two parts, two different little mini talks about different aspects of our research. And so the first one here is about sulfur bacteria and the dissolution of marine seep carbonates. So methane is kind of the simplest and perhaps most abundant organic molecule. It's a colorless, odorless gas, and it's the principal component of natural gas. And it's produced both, there are thermogenic processes that can produce methane, but also biogenic, such as methanogenesis, produces methane. And it's a powerful greenhouse gas, so many people are interested in its role in climate. Lots of methane comes out of various seepage environments that we broadly refer to as methane seeps. And these can be things like destabilized gas hydrates, or they can be things like brine pools, fluid conduits where subsurface fluids are being expelled onto the seafloor in various forms. And so lots of methane is coming out in these environments. Methane is coming out of many marine environments, but lots of it at certain sites that we refer to as methane seeps. And at those methane seep sites, the kind of major sink for methane before it can kind of flux into the water column is a so-called process called the anaerobic oxidation of methane, which is basically a metabolic process that's the product of a consortium, or kind of a synergy of two different groups of organisms. One are methane oxidizing archaea, and the other are sulfate reducing bacteria. And you can see the sulfate reducing bacteria, the green here, and then the archaea, the methane oxidizing archaea, are the red here. And they produce two important products in their metabolism. They're consuming methane, and they're producing bicarbonate, and they're also producing hydrogen sulfide. And so what's the importance of these two products? Well, one of them is that the bicarbonate increases alkalinity in the sediments. So you have carbonate alkalinity gets shifted towards the carbonate ion through this process of the anaerobic oxidation of methane. That causes extensive carbonate, authigenic carbonates to form in the subsurface. So they basically precipitate in sediments at these methane seep settings. You get lots and lots of carbonates, extensive carbonates. You get crusts and boulders and nodules and all kinds of stuff. at many of these methane seep sites. And so you can see this whitish stuff here, that's all methane seep carbonate. And there's this stuff here that's kind of yellowish, I should say, is the carbonate. And then this whitish stuff are actually sulfur oxidizing bacteria. So here are some sulfur oxidizing bacteria, these whitish mats. And these are basically, these are sulfur oxidizers that are fueled by the hydrogen sulfide that's being produced from the anaerobic oxidation of methane. So these are the organisms, these are the sulfur oxidizers here that are oxidizing that hydrogen sulfide that's produced in the AOM, the anaerobic oxidation of methane. And note that they're actually producing acid here. So here's a picture from a submersible of a bunch of carbonate kind of little like chimney type structures here that are all covered with mats of sulfur bacteria. Some of them are yellow and some of them are white, but you can see all these carbonates here, they're all covered with sulfur oxidizers. And so what I should point out is these are carbonates that are basically covered with bacteria who produce acid as a product of their metabolism. So they're basically these sulfur oxidizers, when they're growing on oxygen as an electron acceptor, they're producing sulfuric acid, more or less, right? And interestingly enough, so when I was a postdoc, we did a lot of work with the Alvin submersible, and we collected a lot of these carbonate samples. And I noticed that, and others have noticed this as well, but I noticed that a lot of these sulfur oxidizers, these mats of sulfur bacteria, they kind of co-occur with dissolution features. And so you can see this like kind of ratty appearance of the carbonates here. If you turn that over onto the bottom side and look at the kind of surface that's not exposed to the sulfur bacteria, it doesn't look like that. It's kind of smooth and more or less more smooth. Whereas on the top surface where the sulfur bacteria are, it's all dissolved looking. And you can actually see in the rock record, you can see an ancient seep carbonates. You can see basically kind of what look like dissolution surfaces as well. So there's kind of extensive evidence for dissolution textures that are associated with sulfur oxidizing bacteria. Also, we can see at the micro scale, if we look at like an SEM, we can see kind of a possible association between the presence of filamentous sulfur bacteria and filamentous shaped etch pits on a variety of different kind of scales here. And so what we think is going on is that you have basically kind of two biogeochemical regimes or zones in these seep settings, where you have in the sediments, you have a lot of anaerobic oxidation of methane, and that's causing carbonates to form. And then through typically sediment winnowing, you can have the exposure of calcium carbonates at the sediment water interface, where there you have undersaturated conditions because you have sulfur oxidation occurring there. And so you have these mats of sulfur bacteria that are living on the carbonates, and they're creating a very different kind of geochemical environment, one that promotes dissolution. And so that's what we suggested was happening, but we wanted to test that. This question of, are these acid-producing sulfur bacteria actually dissolving these methane-derived carbonates? And so the first question is, I said that these mats, the whitish mats, are sulfur bacteria, but I just said that because I know that's what they look like. We would have to test that more rigorously. And so we did that. We basically took samples from submersible and extracted the DNA. And then we basically sequenced part of a marker gene. And that marker gene allows us to kind of unambiguously identify a bunch of the microbes that are present in that mat. And sure enough, many of those groups of organisms that are represented in that DNA extraction are known sulfur oxidizing bacteria of many different groups. And so that's just the kind of the results here show that there are many different groups of sulfur oxidizing bacteria that are represented in those biofilms, or those white microbial mats that I showed there. So then we know there are sulfur bacteria there. What if we, in the lab, grow sulfur bacteria on rock chips? Will they dissolve the rocks, and how much will they dissolve them relative to controls where sulfur oxidation isn't occurring? So what we did is we took aragonite rock chips and we put them in a continuous flow bioreactor, and you can see the little chips here. And I should say that I didn't do this; Dalton did this, and it was huge, huge, huge amount of work. And I'll explain more about why in a second. But basically, this took a very, very long time. What we did is some experiments where we have these continuous flow bioreactors where we have very high flow rates of kind of basically what amounts to an artificial seawater, so sea salts plus some growth media, including yeast extract to kind of grow the bacteria. And in some treatments, we had a sulfur source, here thiosulfate, but that's basically operates the same as hydrogen sulfide. It's just a little easier to work with. And then we basically had this all refrigerated, and we simulated saturation states that were the same as what's actually been observed in steep environments. And so then we ran these experiments for basically 3 weeks, or Dalton ran them, which involved lots and lots of changes of the media there, and lots of autoclaving, and it was actually very, very, very challenging. And the pumps were constantly breaking down because it was such high flow rates under cold and wet conditions, and it was really challenging to get these data. But what the data show is basically mass loss of these aragonite rock chips under conditions where sulfur oxidation was occurring versus conditions where the reactors were not inoculated, so they are sterile, or ones where the sulfur bacteria were not provided with a sulfur source, but were instead just growing on the yeast extract. And you can see here there's much greater mass loss of the aragonite chips under sulfur oxidation conditions than there is under abiotic and heterotrophic conditions. And then the other question we want to address is, can sulfur oxidizing bacteria produce acidic conditions under a well-buffered marine environment? So those bioreactors I showed you, we had to do such high flow rates in order to keep them from basically kind of artificially becoming completely acidic, because that wouldn't really simulate what happens in the ocean if you just kind of artificially acidify the whole thing. So those flow rates are based on keeping the kind of bulk fluid at kind of a constant alkalinity that's the same as what we would observe in the seep. And so if you flow enough media through there, you're not like kind of artificially acidifying the whole thing, but rather you're looking at just the acidification conditions that happen within a biofilm. And so in order to kind of show that more clearly, what we did is what Dalton, and here Peter Schrudel, who was working with Dalton, did. He's one of our undergraduates who's now a graduate student in Boston. So we had these little flow cells here. And so the flow cells basically have these carbonate rock chips in them, and then we were able to put them on a confocal microscope and basically image the biofilm. And what Dalton and Peter did is they added a dye, a fluorescent dye that's pH sensitive. And so that pH sensitive dye can allow you to quantitatively kind of measure and image the pH on a very fine scale. So this is a 50 micron scale bar. And so the blue here is actually showing a DNA stain of the biofilm on the rock surface. And then these different colors here on the left are showing the sulfur amended experiments where you have sulfur oxidation occurring, and you can see basically acid is being produced within the biofilm, and so you see basically kind of a pH gradient coming off of this rock chip, and you don't see that pH gradient with the heterotrophic conditions. So you don't see that acid production. So this is basically kind of showing that you can get acid production maintained within a thin, small biofilm that's attached to the carbonate that can basically kind of overcome the bulk fluid acid buffering of a larger system like the ocean and cause acidification that can lead to dissolution of the carbonate. So, but is this dissolution significant to kind of the global carbon cycle? Well, that's a kind of a difficult question to answer because there are lots of unknowns. But we did some kind of estimates and back of the envelope calculations where Dalton looked at a number of seep sites and studies where they have some used sonar and so on to get an idea of kind of the measure of the outcrop at the seafloor. And then Dalton looked at submersible images of carbonates that are covered by these mats of sulfur bacteria, and was able to get an estimate of how much of the carbonates are covered by sulfur bacteria. And then from there, basically kind of extrapolate out these dissolution rates to 10,000 seeps, which is kind of a plausible number for, we don't really know how many seeps are out there, but thousands are known, and get a number like 7 times 10 to the 8 kilograms of carbon per year. And that's quite a bit less than, say, what the kind of dissolution that's occurring if we look at all the like kind of dissolution that is thought to be occurring from anthropogenic acidification of carbonates. But still, it's something. And perhaps maybe there's a lot of uncertainties, but something like 4% of the carbon that gets locked away in this, what was thought to be a geologically kind of stable form as these carbonates, then can get liberated via this mechanism of sulfur oxidation. But as I said, there are big uncertainties on this. We don't know very well the extent of biofilm coverage because we based it on just kind of what was macroscopically visible mats, but there's also mats that wouldn't be visible from a submersible. We also don't know kind of in situ rates of sulfur oxidation. We don't know the surface area of exposed carbonates very well. Also, the composition of carbonate mineral phase is important. So we tested aragonite, but dolomite and calcite are also components of seep carbonates that we would also have to test. And also the total number of seeps globally is still a big unknown. So there's a lot of things we don't know, but we didn't really appreciate before doing this work that sulfur oxidation could contribute to kind of the large scale breakdown of these, or dissolution of these carbonates. Okay, so that's one of the two kind of stories that I want to tell, and then I'm going to kind of spend the second half, or hopefully less than half here, talking about endobionts and Thia Margarita. So now I'm going to switch over from talking about little bacteria to talking about big bacteria. Okay, so in most models of eukaryote evolution, so eukaryotic cells like ours have organelles in them, organelles like mitochondria in us, or chloroplasts in plants, that basically generate energy via aerobic respiration and photosynthesis, respectively. And those organelles are thought to be derived from bacterial symbionts that lived inside of some ancestral eukaryotic host cell, which could have been an archaeon, it could have been something that had many of the eukaryotic features, or it could have been bacterial, probably not bacterial, but it could have been a prokaryote living inside of another prokaryote. And so we have, in the modern biosphere today, we have many examples of eukaryotic cells that have bacterial cells living inside of them. And that's for a number of reasons. There's eukaryotic cells can ingest food particles and bring cells into them in that way. Also, eukaryotic cells are prone to being infected. So things like rickettsia. This is a eukaryotic cell here, and rickettsia is basically these little bacteria that are obligate intercellular parasites of eukaryotic cells. Also, there are a number of symbiotic relationships, like these mussels that live at methane seeps and at methane seeps that actually host sulfur oxidizing bacteria within their tissue, even within their cells. So they're eukaryotic cells that have sulfur oxidizing bacteria living inside of them. But what about bacteria living in other bacteria? Well, that's kind of one of the models for eukaryogenesis, eukaryoevolution. But there are very few examples of bacteria living inside of bacteria in the modern biosphere. There are these mealybugs, which are insects that have in certain cells of the mealybug, there are bacteria living inside of those mealybug cells, and those bacteria themselves have bacteria living inside of them. So it's like this kind of Russian dolls of cells living inside of cells. But in that case, the bacteria aren't free living in the environment. They're actually symbionts themselves of these eukaryotic cells. And there's also, back in the 1990s, there was some electron microscopy results that showed possible cells living inside of filamentous sulfur oxidizing bacteria that are actually interestingly related to our friends Thia Margarita and are relevant to this story. But they were published in the Journal of Microscopy Research and Technique, and they weren't given very much attention, and they didn't show any kind of relationship or any kind of evidence for what these organisms were or what they were doing. So back to our organism, Thia Margarita, our giant sulfur bacteria. These bacteria are so big that they have populations, extensive populations of other cells living on them. So you can see all these filaments here. But if we also went into a kind of higher magnification, there are all sorts of bacteria that are living in kind of a mucus sheath on the outside of this big giant cell. And in that way, we think that these organisms have a microbiome that's sort of analogous to the microbiome that we have on our skin and in our gut, that we're now increasingly recognizing that are important and in our interactions and in our human health, right? And so we were aware that these bacteria, these giant Thia Margarita, have bacteria living on the outside. But what we later discovered, we spend a lot of time trying to grow Thia Margarita and work with them and do various things, experiments with them. What we discovered is basically that there are cells living also are found inside of Thia Margarita. And so you can click, if you see this movie here, all those little structures that are moving around in there, we observe those in many of the Thia Margarita cells that we have. And so these led to a number of hypotheses. Maybe what are these things? Maybe they're giant viruses. Maybe the host Thia Margarita is actually dead and some bacteria are just getting in there, eating it or whatever. Or maybe there's some kind of spores of Thia Margarita. Or maybe there are actually some endobionts, so cells living inside of Thia Margarita. And so we set out to kind of test that idea. And first off, we wanted to test whether or not the endobionts, so the cells inside the cells, are alive, and also whether the host organism was alive, the host Thia Margarita. And so what you see here is actually a so-called live dead stain that's basically indicating the active metabolism of the Thia Margarita cell, which is represented by this kind of thin line here. And then all those little green dots inside, those are the endobiont cells. And so this is a confocal microscopy, so it's illuminating just a very thin plane of the cell here. And so that's kind of why it looks the way that it does, but it's allowing us to kind of see inside of the intact living Thia Margarita cell here. And we also needed to demonstrate, that these organisms were really alive. So the confocal microscope does that, but also using a transmission electron microscopy and cryofib electron microscopy, we were able to basically image these endobionts inside of Thia Margarita. So Thia Margarita is actually really, really challenging to work with electron microscopy for a number of reasons. The biggest one is because it's basically like a water balloon. It's got this huge vacuole that's full of water, and it doesn't behave well when you fix it for electron microscopy, and bad things happen. And so we've spent years basically trying to get good images, and so these are some of the examples of what we've done cryofib, and also transmission electron microscopy. And so this is the outside of the Thia margarita cell, and this is kind of the margin of Thia margarita, and here's its cytoplasm, and here's the vacuole, and then all these little dark structures are the endobionts. And we used a bunch of other lines of evidence basically to indicate that these Thia margarita cells and the endobionts living inside of them are actually metabolically active. So there's a whole bunch of different dyes, details of which I don't need to get into, but dyes that show basically active metabolism. Here's one that's basically showing the incorporation of fluorescently labeled amino acids. So that these organisms are taking up these amino acids and incorporating them, as are the Thia margarita cells. And then also we see apparent evidence of inheritance of the endobionts across division planes. So we can't grow these bacteria in the lab, but we can keep them alive for a long time, up to years, and we can take them out of their sediments and see evidence that they're in the process of dividing. And in cases like this, what this appears to show is that you have this cloud of endobionts that are represented here by these kind of lighter speckles. The darker ones out here are actually inclusion bodies, but these lighter ones are the endobionts. And you can see kind of a mirroring of this cloud across this division plane, which suggests that they maybe were inherited. The daughter cells are inheriting this population of endobionts during cell division. Okay, so it seems that there are cells living inside of Thia margarita, but who are they? Well, this took us a long time to try to figure out, but we think we have an answer of at least some of the players here. And interestingly enough, if maybe you've seen videos or otherwise familiar of these tube worms that live at hydrothermal vents at black smokers, you see there's massive populations of these tube worms. And these tube worms basically make a living by having sulfur oxidizing bacteria inside of them that grow in this special little organ. And they basically kind of farm these small sulfur oxidizing bacteria. And interestingly enough, our endobionts inside of Thia margarita, when we look at their 16S marker gene, their most close relatives are actually the symbionts of these tube worms, which is quite interesting. And it makes sense because these endosymbionts of tube worms already have this kind of endobiont lifestyle. But it also leaves open the interesting possibility that this kind of association of these sulfur oxidizing bacterial symbionts predates animal life, right? Maybe the relationship of them with Thia margarita or other organisms actually came before their relationship with tube worms. That's one possibility that's quite interesting. And we wanted to show kind of more rigorously, in addition to kind of pulling out individual endobiont cells and sequencing the DNA there, we also were able to make fluorescent probes based on those results that are specific to the probes specifically bind to the DNA of that particular group. So we designed those probes, and then we used those to go in and probe in situ the location of those the sequences or that are bound to by those specific probes that we designed to bind to just the endobionts. And so here is kind of a video of confocal microscopy that shows the edge of the Thia margarita cell that we're zooming in on here. And so here in red are basically the endobiont signals, the probe basically that's binding to the endobionts. And we can see that it localizes to within the margin of the Thia margarita cell here. And so this is basically like a maximum intensity production of a three-dimensional collection of slices of confocal images that you're seeing here. And finally, we wanted to have get an idea of what these organisms are doing. How are they interacting with Thia margarita? And in order to do that, we we've tried many things, but we tried to culture them and so on, so far without success. But one thing we were able to do is extract out all the DNA and sequence all the DNA from the entire Thia Margarita and the bacteria that's associated with it. And then we're able to basically pull out, using bioinformatics, which is what Beverly spends most of her time doing these days, basically pulling out these different assemblages of sequences and kind of binning them. There are different properties you can use to basically kind of take all those little fragments of DNA and kind of put them into bins that represent the genetic contribution of different organisms. But this requires a lot of basically supercomputing computation to do. And then you can go in and kind of analyze the genes and look at what those different organisms in that population have the genetic potential to do. We can't say that they're doing that, but we could say what they have the genetic potential to do. So what are the endobionts doing? Well, interestingly, they have, just like Thia Margarita, they use nitrate as an electron acceptor and sulfur as an electron donor. And just like Thia Margarita, they can fix carbon via the Calvin cycle, just like plants. Also, unlike Thia Margarita, they have genes for these things that are called secretion systems. And secretion systems are basically like a little bacterial syringe, in a way, that is, it's like a little machine that basically, kind of a biochemical machine that exports proteins. And so you can use those proteins to shoot proteins out into the extracellular environment or into other cells. And so they're often used by pathogens in their infection of other cells, or in their virulence or disease causation. But they're also used for a variety of different other things. And so this is interesting. These, based on these kind of results, their metabolisms are pretty similar in a lot of ways to the host Thia Margarita cell, and especially in that they're using nitrate as their electron acceptor. So it could be that they're actually parasites that are stealing vascular nitrate. So you've got this big scuba tank of nitrate inside Thia Margarita, and imagine that you're one of these endobionts, and you're living out there in the system that actually goes anoxic periodically, and maybe then you don't have anything to breathe. But if you can get inside of Thia Margarita, there's a bunch of nitrate in there, so maybe you could use that and basically kind of steal it. So that's one possibility. We don't know that for sure, but that's something that we're considering. So there's still a lot of open questions here. Are the endobionts interacting with Thia Margarita? If so, how? How are the endobionts getting inside of Thia Margarita? Are the endobionts influencing the whole environment? So Thia Margarita is known to have a whole bunch of impacts on the biogeochemistry of the sediments, including they're important in oxidizing sulfide that would otherwise get to the water column and do things like kill fish, which actually still happens under sometimes anyway. But organisms like Thia Margarita are kind of modulating that sulfur that gets out into the water column. Also, Thia Margarita is involved in things like the formation of phosphorites, so calcium phosphate mineral deposits. We don't know if these endobionts could be somehow involved in that. We really don't know what their biogeochemical significance is. And we also don't know if they have some evolutionary significance. So we're not saying that these endobionts living in Thia Margarita are analogs for the early eukaryotes, but we have very few systems, basically almost none, of free living bacteria that have bacteria living inside of them to study, to ask questions about, like, how do bacteria even infect other bacteria? And so these kind of questions we might be able to learn something from in this system that might have kind of implications for our understanding of early eukaryote cells. Before I end my talk, I just want to thank the RGNO Namibia course. This is a course that we hold every year, except for during COVID. We haven't been doing it, but for the last 7 or so years, we've been doing this course in Namibia, where we basically have students come from all over all over the world, including about half the students come from Namibia or other countries in Africa. And basically, we have this cohort of graduate students that does research on the upwelling system. And some of those students do, they have diverse kind of projects that they end up doing. A lot of them are biology focused, but not all of them. Some of them are more kind of geologically focused. But they're all focused on the Benguela upwelling system, which is that upwelling system off of Namibia there. And as part of that course, we are able to use the research vessel Mirabilis, which is Namibia's research vessel. And they have a great crew and scientists of the Ministry of Fisheries there that come in and they do their own science on the ship, but they also work with our students in the course. And together we mentor the students to do research off of Namibia there. And we do that every year for several weeks. It's a regular, recurring kind of program. And here's a picture of Dalton, who was a participant in the course as well. And here we are basically with a core that we collected with the Mirabilis. And this is where we are collecting the Thia Margarita here. Okay, and I just also want to thank the National Science Foundation, Simons Foundation, and NASA for supporting this work, and the University of Minnesota. And thank all of you for attending and for all of your support over the years.
Thanks, Jake. As I said at the start, we'll open it up for questions from students first. And you can either open your mic and ask your question, or you can type it out in the chat and I'll read it out. Anyone?
Hey Jake, this is Jen Taylor. My question is kind of among the Thia Margarita, do all of them have endobionts, or do you see some variety? Do some like not have endobionts, or are they all kind of uniform in that sense?
That's a great question. So if you'd asked me that question a couple years ago, I would have said that maybe about 15% or 20% of them have the endobionts and the rest don't. But as we've gotten better at our microscopy techniques, we've basically started to observe that a lot, most, maybe all actually have them, but just not very high accumulations of them. And you have to use pretty, like if you're using like sensitive kind of confocal techniques, then you can start to see like, this cell has a few of them, even though under like other less sensitive microscopy techniques that you'd use to kind of screen them, you wouldn't be able to see those at all. And so probably most of them seem like they have them. I think that's basically kind of the conclusion that we're coming to, but we don't know why. Some of them have these like really, dense accumulations and basically like big clouds of them that are kind of filling up the cell, and others have very few, just like literally like a dozen or something. But we don't know why.
Other student questions?
Hey Jake, this is Chris Schuler. I have a question about the seep carbonates. Do you know what the speciation of carbonates are like in most of those methane seeps? And do you have like a theory as to how it would affect dissolution rates generally?
Can you repeat that one more time? Sorry, it kind of broke up on my end.
Yeah. Do you know what the speciation of carbonates are like in those methane seeps? And do you have like a theory as to how it might affect dissolution rates?
Oh, the speciation, like chemical speciation of like.
Yeah, like is it mostly aragonite versus like dolomite or like that?
Yeah. Okay, so seep carbonates are kind of complex and heterogeneous, but a lot of them, if you look at them under like in thin section or something, the in many cases, they're like basically a bunch of like aragonite crystal fans that are basically kind of precipitated in kind of spaces, void spaces in the sediments, more or less. But there is some calcite that's present as well, and then also in some seep sites there's dolomite as well, and so all those have different KSPs. Aragonite would be the most susceptible to dissolution, and then calcite, and then dolomite the least. Dolomite usually, it's kind of complicated how dolomite actually forms in these seep systems, and I think not fully understood, at least not by me. And they're oftentimes like kind of found at the margins of the seeps, like the dolomite. So there's probably some kind of diagenetic process or something that's resulting in dolomites at seep environments. But there's also thought to be authigenic dolomite as well that's influenced by bacterial sulfate reduction. But yeah, definitely the different dolomite would be much less susceptible to dissolution than say aragonite, for sure. But there's a lot of aragonite in methane seep carbonates.
Other questions from students? Questions from other students?
Actually, here's a seep carbonate right here, and you can see how like kind of complex it is. And there's a lot of like shell material and stuff that's like kind of welded together too, in addition to the actual like aragonite, authigenic aragonite cement that's like welding it together.
If no other student questions or questions from other students, we'll open up to anybody who has a question. Christian says, following on Jen's question, does Thia Margarita contain a certain quantity of endobionts, or does it show cycles with more or less guests, depending on environmental or other changes?
We aren't able to answer that question, to be honest with you, because it's we basically collect them. We have maybe if we're lucky, we have an opportunity to collect them once a year, going all the way to Namibia and collect these from if we're lucky, a few different sites there. And we do try to characterize the geochemistry of the sites where we collect them, so far we don't see any kind of real. There are certain sites that seem like they have much more dense endobionts when we collect them than others. We're not exactly sure why that is, but there's actually quite heterogeneous kind of geochemical conditions at the sediment water interface at different sites. And in fact, you even have different large sulfur bacteria that can be found like in heterogeneous populations, like patches on the seafloor there. So you might have Thia Margarita here, and then you might have Vegeatoa, which are like the cousins of Thia Margarita over here. And why one lives here and one lives here probably has something to do with very fine scale geochemical differences in oceanographic currents and organic matter load and stuff like that, which are very difficult to actually measure. There's one kind of benthic lander at one of these sites that's measuring oxygen, but we can't. It's very difficult to like compare site to site. So yeah, I don't know the answer to that. It seems like there are some sites that definitely have more endobionts than others, though. So there is something going on there, but we don't know what.
So Jake Michael Chen asks, do you have a sense for why the sulfur bacteria are colonizing carbonates, or are they colonizing solid surfaces in general and carbonates just happen to be common?
That's a really interesting question, and a good question. We did some initial kind of experiments to show if there's like preferential kind of attachment, and it's not kind of clear whether there is or not. But one possibility that we thought about early on was that, or we still think about, is that The marine environment isn't really thought of as being carbon limited because there's a lot of DIC in the ocean, right? But there are maybe environments where you could have carbon limitation under kind of microenvironments. And one of those might be within a biofilm. So maybe at the bottom of a biofilm that's attached to a carbonate, you could have a carbon limited environment. And these organisms are fixing CO2. And so maybe you could have, through the dissolution of that carbonate, you could actually have carbon that's provided. Also, these little sulfur oxidizing bacteria, they live in these like white smoker environments. And they're actually encounter carbon limited environments as well. And so they live on carbonates in those that are carbon limited in white smoker settings. And so that's another place where they might actually benefit from the liberation of inorganic carbon from the carbonates.
So Jake, I have a question for you. You've been telling us since 2010, if not before, really, with your publication on the Shantu microfossils, that Thia margarita is the largest bacterium in the world. Now, is that really true, or are they cheating because they've got the vacuoles? And so my question is, do they have the same amount of, do they have more cytoplasm than any other and associated contents than any other bacteria, or are they just cytoplasm stretched over a giant internal structure?
That's a great question. Actually, I would say that they're the second largest in terms of cytoplasm volume. There's a organism called Epulopycium. Epulopycium, it's a genus that basically are very large bacteria that are on the order of, I think, 300 or 400 microns in the largest ones that live inside of parrotfish guts. And they actually have this really like convoluted kind of membranes and stuff that are in there that may allow them to kind of get around this question of like surface area to volume, which is what really kind of inhibits bacteria from getting big usually. And so that's one possible kind of that's a candidate for really the largest bacterium is without cheating is this
Right.
Epulopycium.
Well, that raises the other question I had, which is that you often talk about the vacuole, say with regard to nitrate being like a scuba tank, and they've got this store that they can use as needed depending on environmental conditions. But could it be, in fact, that given their volume, given the surface to volume area issue, that diffusion of oxidants in from the exterior may not be as efficient as being able to store it and having it diffused from the interior?
Well,
or is the scale not so great that it matters really?
I think one problem with that is that I think they have to expend, and they may have to spend energy to get the nitrate into the vacuole in the first place, because they have to transport it through multiple membranes.
Right.
And it doesn't just go in there. I think they have to actively transport it. So I think in that sense that they're like actually spending energy under one kind of metabolic regime to store it for a later time.
Right. Cool.
Yeah. But actually, that thing about the cytoplasm, I just want to say real quick. Dalton actually, more recently, he's been doing a lot of this like 3D reconstructions of the imaging of the cells, and some of them actually seem to have a lot more cytoplasm than we thought before. Like that image I showed before that shows the vacuole in there. There's actually a lot of cytoplasm in that particular Thia margarita, much more than we thought before. And so that's actually kind of something that's just come out of his more recent imaging, which is pretty cool.
Are there other questions for Jake?
Or if I can chime in real quick, Jake, too. Some of them have like a diverse, or like rather than having like one very large central vacuole, it seems through imaging that there's many smaller vacuoles within there too, which kind of complicates things even further.
They're very interesting organisms that we're learning more about all the time.
As it should be.
Yeah. Thanks, Tom.
All right, well, if there are no other questions, remind you that you're welcome to gather up on the west side of Tate for a little hangout if you're nearby and have time. And otherwise, why don't we thank Jake for his stimulating presentation, and we'll see you all again.
Thanks, David. Thanks, all.